Okay, assuming a DNA replication in this particular bacteria occurred in a similar mechanism to humans, state and describe the mode of DNA replication. So, I just want you to describe what semi-conservative replication is. Okay, so that's what the question is asking for. Okay, usual, if you found the question challenging or you learned something from it, please star it, include it in your notes. This is not the end game. The end game is your end of year or your mid-year. Okay? No lah. One student told me the end game is learning for life. Okay, you win. Okay, next question. Draw the possible observation for DNA bands. This question is a test of whether you understand which part. The semi-conservative, A part 2. Yes, semi-conservative. The observations you expect from Mesosen and Style experiment. Okay, A part 2 is assessing your understanding of Mesosen and Style experiment. Okay, next, I'll go through answers a while. 1B, name 3 enzymes required for DNA replication and describe their functions. What's this question asking for? As it is. Yeah? Exactly what you're asking for. I think this question is very generous because there are many enzymes involved. Just need to pick 3. Okay, C. Okay, C, Let me guess what this question. Suggest why replication only occurs in the 5 to 3' direction. Okay, 8 before 3 at 5'. 5 to 3, Okay, so they ask you why. Okay, so they're asking you 3', 5'. They're asking you how come you can only add new nucleotides always to the 3, to the 3, to the 3. Okay, why can't we add it on the 5 end? Why can't we elongate this direction? Why only elongate in this direction? 5 to 3. Okay, why? What do you think this question is asking?
Because the anchor is placed on the 3 end. The driving nucleotide anchor is based on 3 end. And since DNA is anti-parallel, the DNA strands are anti-parallel, needs to be elongated from the
Oh. But nothing's stopping me from placing a primer at the other end.
But there's an anchor.
Anti-parallel?
The anchor is on the 3 end.
Yeah, so if you mention something along the lines of adding in the 3' OH, you have gotten some up there. Okay? Actually, this question is asking you a JH3 topic. Why can replication only occur in this direction? Okay, it's a JH3 topic. If I tell you the topic is enzymes, will you be able to answer it? Okay, so we come to it in a while. Okay, D, question D, all they're asking for is complementary base pairing. If you understand the idea of it, but in a very complicated way. Okay, they give you a lot of things to calculate. Last one, question 2. This will kill me for this. Question 2, DNA packaging. They just want you to compare, contrast DNA packaging and DNA that's not fully packaged. Sorry, okay, I'll give you 2 mark, 50 mark. Thank you very much. Okay, ready? Okay. Question B. Can you take 1A part 2? Is this what you do? Masih label to me. Just put like that. I'm going to be thinking about is this. Your upper band, make sure it's aligned to 2A's upper band. Okay? Some of you made it thicker, but that's fine. As long as it's aligned with that region. Okay, 1A part 1, semi-conservative replication. This literally like from your RE. Okay, the first point is 1 mark. The other 2 points, either or, give you 1 mark. Yeah, if you find a question challenging, make sure it's star it. Or if you learn something new, they want to put in your notes for mid-years.
Can I just use one of the 2 points?
Yeah. Just either or. Okay, could you use parental strand, daughter strand? Okay, instead of the what I used the first time on that side, the original and new. Okay, daughter and parent. No. Sorry. So what's the best answer? But you think we say synthesize. But it's fine. Okay, can I move on? Yeah. Ashley?
Yeah.
Oh, okay. Instead of semi-conservative, no. Instead of semi-conservative model, you put semi-conservative replication. Okay. Yeah. Yes. Okay, name 3 enzymes. Okay, what are 3 enzymes? Oh, there are so many. Yes. Yeah, can. Okay. But this time, right, you can model or replication. Okay. Next, 3 enzymes. I hope you can get 2 marks for this stuff. After you make notes for this, right? After this one part of the notes you should. Right? Okay. So any 3 of the enzymes. Any other enzyme you have? Maybe you had the other DNA polymerase. Yeah. Okay. That's important. Okay, so when you talk about DNA polymerase, it's important for you to describe where you add the nucleotides to. Alright, you add it to the 3' OH. And then you synthesize in a 5 to 3. If it's not your notes, can you make sure it's in your notes? Yeah? We add the 3 deoxyribonucleotides to the 3' OH end. Okay? In other words, you're synthesizing in a 5 to 3 direction. Yeah?
Wait, the DNA polymerase is under the same point. There's 2 like.
Yeah, there are 2 different kinds. So actually, you should separate out the 2. Yeah. Do you write about the other 2 or not?
I know about like the one that adds the. But I say like the catalyzing formation.
Or the one that swaps out primer, is it? Or the other one that is okay. There are different polymerases. You have a question here? Yeah, bio, we don't have a negative coupling. Like if you see something wrong, we won't like minus it. We will look for the good in you. Okay? Yeah. But you know what? DNA primase is its goal is not to elongate.
Yeah. I just added where the.
Oh, okay. Yeah. That's not wrong. It's just a function of DNA. Yes. Okay. Suggest why replication only occurs in 5 to 3' direction. Okay, it's not very hard. Don't see how you can. Okay, this question is a question on enzymes. They are asking you essentially, how come this enzyme can only function in this way? Anti-parallel? No, it's to do with like active sites and stuff. Specific action. Okay, but I'm sure you can get the last one, right? Yeah, at least get 1 mark for this. Okay, this one's still reasonable. Yeah, the first one is also still reasonable. DNA polymerase, specific action. Oh, what's DNTP, is it? Deoxyribonucleotide. No, don't write that. Write the full one. There are phosphate groups in the deoxyribonucleotide. Okay, 1 mark.
If you write anti-parallel, that's also correct.
Are you saying because the template you read
Yeah, I wrote the dentistry is in the
and then that's why you synthesize it in the 5 to 3? But then why can't we read it, right? Okay, the fact is that we read it from 3 to 5, right? And therefore we synthesize 5 to 3, right? So it's the same thing. Then why can't we read from 5 to 3 and synthesize it in a 3 to 5 paragraph? Right? Are we answering the question?
But then now you assume that you know you're already reading it in the 3 plus 5 paragraph. So that's only possible for
Okay, then which one came first? Is it because we read it 3 to 5, therefore we synthesize in 5 to 3? Is it because we read it in 3 to 5, that's why we synthesize in 5 to 3? Or is it because the enzymes that we have can only do it in that direction, therefore we read it in 3 to 5?
How do you know that?
We don't. There is. Oh, say again. How do you know? This one is suggest bio. This one is suggest.
So we can suggest something wrong.
So you can suggest something logical, and if it's logical and it's correct, then you're correct.
The answer is logical.
Okay, what's your answer?
The answer.
Why you laugh? I thought it's logical.
No, but the answer is that the anchor is based on the 3 prime end, so that of the parent strand. Yeah. So since the anchor is on the 3 prime end of the parent DNA, the DNA needs to be read from the 3 prime end to the 5 prime end, and it needs to be synthesized from the 5 prime end to the 3 prime end direction of the newly synthesized.
But what I told you is because the enzyme can only do it in this direction, that's why we put it in this end.
How do I know?
If not, I can put it here. So we're not going answering the root of the issue here. Let me think. Am I being recorded? But it's not here over there.
If they ask me this question again, I'll okay.
Yeah, true. Yeah, that's why it's not obvious. We won't be so cruel. We won't be so cruel. Okay, got it? Yeah? It's not being recorded, but just nice of this sound. We won't be so cruel. Okay, the next question. Hey, whose car is that? Hey, the whole day is like that. Wow, that's so okay, next question. Wow, this question, just now as I was standing there, I was like thinking to myself, how to calculate. I thought very hard also, okay. Okay, the only thing we're testing here is complementary base pairing. That means if I have 2 A's, we need 2 T's up. If I have 2 C's, we need 2 G's. Okay, so how do we complete this up? Okay, plus minus strand. Plus minus strand just means either of the other strand lah. Okay, there are two strands of double stranded DNA. Okay. Okay, so how to calculate? Maybe the first thing is this. What the? Okay, AGCT. Okay, which one goes with which? A and T, C and G. Right? Hello. A and T, C and G. Okay, so next, if they say 20% of the DNA is G, 20% is C. Right? It needs to match up. So therefore, if there's 30% T, then 30% A. Okay, so this one's okay. Okay, next. Strand number 1 and then strand number 2, the plus and minus. Okay, so it says that there's 15, 25, 15, 28, and 32. So how to do this? Okay, you find what's complementary to each other. If there's 15 G's, there should be 15 C's. If there is 25 C's here, there should be 25 G's. If there's 28 C's, there should be 28 A's. If there's 32 A's, there should be 32 T's on this strand. Okay? Okay, then how to calculate this? Hello. You're like the car already. Okay, so how to calculate this now? Ratio means you take one divided by the other. Okay, so I give you an example. Ratio of A plus G to C plus D. What I mean by that is ratio of A plus G divided by C plus T. So this is 50 divided by 50. That's how they give you 1.00. Okay. So how do you get to this next ratio? You take 28 plus 15, 25 plus 32, then you do the math. Don't know what math is. 43 divided by 57. And then you will get 0.74 something. Then you round up to 75. So going by that logic, what is the answer for this? Ratio of A plus T. A plus T. A plus T is 60 divided by G plus T, 40. And you get 1.5. Okay? Wow, this one, testing math, not complementary based, right? Oh yeah, so how do you get marks for this, right? Okay. This is 2 marks. What 2 marks if all values correct, and 1 mark if there's 1 mistake. Wow, brutal. Okay. 2 marks if all correct, 1 mark if there's 1 mistake. Wow, this is brutal. Okay. Sorry? 2 mistakes, 0. Okay, so you did it. I see how I feel when I'm up. Okay, last one. Okay, with reference to eukaryotic chromosome. Yeah, I say it's before, what's eukaryote? Prokaryote. Karyote means nut. That's it. Nut. Okay, so I'm going to find out. Karyote means nut in Greek. So eukaryote means there's a nut. Prokaryote means no nut. So what's the nut? The nut is the nucleus. That's what they observe. Okay, so eukaryote means this organism has a nucleus. Prokaryote means no nucleus. Okay, got nut, no nut. Compare the level of DNA packaging in a dividing and undividing cell. Okay, at this point I'm going to draw the cell cycle again. you may want to take note of this so that you are aware of the various levels of packaging at different points of the cell cycle. Okay, some of you may already have this diagram. More complex circle. Yeah. Okay. So here 1, 2, 3, 4. Okay. We will talk about this tomorrow. M phase. Okay, you will learn that there are various phases. There's prophase, there's meta, there's ana. There's an ana phase. And then there's a telophase. Wow, this is dedicated to you too. Okay, so then there is G2, there's S, and then there's G1. Okay, what level of packaging exists at various parts of the cycle? Firstly, in this part of the cycle, you will find all your DNA in the form of chromatin fiber. Yeah? After we go through the S phase, you will find that the DNA is still in chromatin fiber form, except now we have two conjoined at the centromere. So this is the centromere, and we have two chromatins attached to each other. Okay, but they are sister chromatids now. Okay? Then at this phase over here, prophase, which is called the M phase, you will find that all these two sister chromatids will condense, and condense to a single, and it's connected to a one point, still the centromere. Okay, so the question is asking, with reference to the eukaryotic chromosome, compare the level of DNA packaging in a dividing and undividing cell. So if a cell is not dividing, you will find the DNA in this form, chromatin fibers. If it's about to divide, you will find it in this form, in metaphase chromosome form. Okay, so they just want to know the difference. So what is the similarity? Either or, there will be nucleosomes present. That's the similarity. Whether it's chromatin fiber or chromosome, there's still nucleosomes present. Except for chromosomes, we condense even further.
One mark for similarity, one mark for difference.
Yes. Yes.
So the difference is you see DNA packaging and dividing cell at the chromosomal level, whereas DNA packaging in an undividing cell is at double stranded structural level. It's like I forgot that it's a case.
It's in chromosomal level.
Whereas the undivided cell is at double stranded structural level.
No, I've never seen this way of describing it. Then undivided double stranded structure. Okay. I see, you understand, but you know, I cannot use you because it's not, I couldn't grasp it. It's not a standard way. So, unfortunately, I can't do this. It's not how biology would describe it. Yes. Oh, okay. Akio asks, what are examples of cells that don't really divide anymore? Your brain cells. No, this is legit, you know, so you have an issue here. Don't go and whack your head on the wall or something, because these cells, right, you know, whatever you have there, that's it. So don't go and damage it. Yeah, don't go and let me see. No. The cells will divide, but they'll reach a point where the cells will go up. So there's actually a phase called G0. G0 is once you enter it, you're no longer going to divide anymore. Okay, a lot of your neurons you enter G0. Okay, that means they're not going to divide anymore. Yeah. Okay. Wait, let me hold on there, Kavish. Okay, what? Same. Okay, let's link it together. Then, Jian and Kavish asked, right? Okay, so this is a neuron that doesn't divide anymore. And Jian asked about cancer, right? What's cancer? What is cancer? Cancer is a cell that's going through cell cycle multiple times, out of control already. Okay? On a normal basis, you want to regulate. I only want extra cells when I really need it. Cancerous growth is when the cell just goes out of control and it just creates many copies of the cells. Then it ends up becoming a tumor. So the question is, is the question that your brain will get bigger if you have tumor?
So how does it
Oh my god.
Okay, it's true that you have more brain cells as a result of cancer. However, these brain cells aren't perfect copies of each other. When you go through cell cycle too fast, there will be mistakes made. Okay? On a normal cell cycle basis, actually, when you go to SH level, there are multiple checkpoints along the way to check whether you divided correctly. For example, after you replicate DNA, the cell will check. Did you go and put the correct A with the T and C with the G? They'll check through first before you go on to the next phase. But when you replicate too fast, they skip all these checkpoints. The resultant cell that comes out that's in the tumor is a mutated cell. It cannot carry out its original function. It just exists as a large lump of tumor. Yeah?
So the thing is, no cells don't divide at all. So how do they
Oh, okay. So it would be those cells that, okay. So you're asking if the neurons don't divide anymore, then how come you can still have cancer? Not all the neurons are not dividing. So some can still divide. Yeah. Okay. Okay, for this particular topic, which JS3 topics you can link to? Okay, so now we know what? Enzymes, because of all the DNA replication, helicase, DNA polymerase, so many things, enzymes. One more would be biomolecules, more specifically nucleic acids. Okay, so what are your topics for mid-years? Okay, what are the topics for mid-years? J3, J4. J3, biomolecules, enzymes, membranes, and cells. Okay? So I don't need all the homeostasis on there. Cannot? Okay? Okay, expect the questions to be infused or separate. Okay? So must be keep your mind open. Right? Notice all our quizzes, right? We sneak in some J3 question here and there. Okay? So what are the topics? We know. Okay, your chapter stack, you make some notes, you revise for to a certain extent. Okay, respiratory. Okay, chapter stack done. Okay, energetics. This is respiration plus photosynthesis. Okay. Then what? Oh, then you're here already, right? A cell cycle. What comprises the cell cycle? The S phase, which is your DNA replication plus experiments. Okay, two things. We've just done a chapter stack for that. Okay, then the next one is mitosis. This one, I do have a quiz, but it's a mixture of mitosis and meiosis. So I'll give you the quiz, but as practice, as practice. This is the only thing that I've not asked for a chapter stack for. Okay, so you may want to make it yourself. Okay, so these are the topics. Expect all these fellas to be infused somewhere inside. Yes.
The cells include immunology.
Sorry?
The cells include immunology.
Oh, the cells include immunology, no? Okay, so your book 2 of JH3, actually that one you can.
Not okay.
No, put on your pillow, sleep on it. Okay? So you book 1, right? 1. Okay. How do you all feel? How do you all feel about it? Because it's in week 7.
Actually, I'm studying it, so yeah. I'm quite relaxed.
Okay, I heard that bio is same there as the humanities, right? Right or not? So if you take geography, it's going to be geography. If you take history, it's with history. Okay? These two are like, it's not the best combi to have on the same day. Okay? Yeah, so for biology, I hope that this time around, for the first time, that since you'll be making notes along the way, it won't be so painful at this period of time. It was painful for you for the past few months, but I'm hoping that for you now, it's less painful for biology. Okay? Yeah, biology is admittedly content heavy, but that's why we'll be trying to be consistent, and I thank all of you for trying your best. Okay? So we have 1 week of revision next week, right? This week we are going to complete mitosis. It's not very difficult. Okay, mitosis. 1, 2, you can go and read up already. Page 90. Page 90 to 90, 95. Okay, up to 95. If you want to be done with the chapter yourself, right, don't wait for me. You want to complete this week already, or today or tomorrow, 95 is the end. Okay? If not, you can wait. And then this Thursday, I'll meet you again. Alright, time's up, right? Okay, thank you. Have a good day. See you again.